Hello, and thank you for joining today's Environment and Energy Leader webinar titled Successful Use of Pilot Testing to Address Evolving PFAS Treatment Regulations Around the World, sponsored by ECT2 of Montrose Environmental Group. My name is Jessica Hunt, and I'm the Director of Live Events for E&E Leader. We appreciate everyone joining us live, and a large thank you in advance to those who will be watching this on demand. I will apologize in advance if you hear my son in the background during today's event. Before I hand the presentation over to our speaker, I do want to go over a few housekeeping items. If you experience technical difficulties during the event, please follow the troubleshooting tips located in the help widget at the bottom of your console. If you are still having issues after following the steps, please send a message via the Q&A widget, which is open on the top right part of your screen. Please take a look at the list of resources today's sponsor has provided for all registrants. Once the event is archived, you will be able to jump back onto the attendee console in order to download a PDF copy of today's presentation. We will be holding a Q&A session at the conclusion of Steve's event today, so please submit questions over the next 45 minutes or so. We will do our best to try to address as many as possible during the Q&A, but if we do not get a chance to answer your question during the live event, the speaker or a member of his team will follow up with you as soon as possible. I am pleased to welcome Steve Woodard from ECT2. Steve has over 3 decades of experience in water and wastewater treatment and currently holds the position of SVP of Research and Development at ECT2. He holds 10 patents for technology developed in the implementation of various systems and methods for treating water, wastewater, groundwater, and vapor. Steve has also authored multiple publications, including journal articles and textbook chapters related to his industry knowledge and expertise, and has a PhD in environmental engineering from Purdue University. At this point, I'm going to turn the presentation over to you, Steve.
All right, great. Thank you, Jessica, and thanks everybody for joining. I think we're going to have some fun today with this one. It's a pretty cool topic, and I'm going to keep it pretty loose and share some of our company, ECT2's PFAS treatment pilot testing experience from a global perspective. So lots of good case studies. The next slide, presentation outline. So this is what I'm going to cover. I think it's important to start with an overview of the regulatory standards, at least for the parts of the world that are actively developing and enforcing standards and guidelines. North America, Western Europe, and Australia. And we'll look at what are the options? What are the proven technologies? What should we be looking at piloting? Then I'll discuss the importance of proper pilot study design, why pretreatment is a critical component, and also don't forget about the data. Data collection and interpretation is often underrepresented and does not typically lead to positive outcomes. We're going to focus some on that. Scaling up for success. The importance of proper scaling is important, and then we'll dig into the case studies, which I think you'll find interesting. So the next slide, ITRC has a lot of you are probably familiar with this organization, and the link is included here, but they have a good website that provides information on PFAS regulations, not just in the United States, but also in Canada, Australia, and in Europe. So that's worth checking out. Next slide, summary of PFAS regulatory standards. About half the states in the U.S. have published standards or guidance values for PFAS compounds. And not surprisingly, PFAS and PFOA lead the pack as the most regulated compounds. I've listed down below California, Michigan, New York, New Jersey, some examples of states that are either already below single-digit nanogram per liter or part per trillion levels, or are headed in that direction. And it's pretty amazing if you think about where we were just 4 or 5 years ago. Things are really progressing. And if you look at New York State, they have designated PFAS, PFOA as hazardous substances, which gives the state the authority to enforce cleanup of sites that are contaminated because they're with PFAS because they're deemed hazardous. New Jersey has promulgated MCLs for PFAS and PFOA. So the states are beginning to promulgate MCLs, which is a big deal. They're also beginning to designate PFAS as hazardous substance, which forces the cleanup. And then there's a bill, the PFAS Action Act. I believe it's on the Senate floor now. But over the next 2 years, Congress will be requiring EPA to do these things at the federal level, the forcing EPA to promulgate MCLs for PFAS and PFOA, designate PFAS as a hazardous substance, and even hazardous air pollutant designation to crack down on incineration. So lots going on the regulatory front in the states. And if you look at the next slide, which shows the drinking water regulations in the U.S. as of about a year ago, exactly a year ago, you can see that short chains are starting to pop up. There are states in dark blue that already have promulgated values that are law. Starting to see the sum of multiple compounds, like 5 or 6 or 7 compounds, having to be lower than a certain level. And then clicking to the next slide shows the map nearly a year later, June 2021. So this is very current. This is where we're at now. And what we see are more states are promulgating laws, more compounds are being regulated, more stringent regulatory levels are being applied. So things are really on the move from a regulatory standpoint with PFAS, and it's going to be very interesting to see what happens in the coming months and years. So the next slide, we get into the global situation a bit. Australia, Canada, the European nations, they are at it as well. And if you look at Sweden, for instance, their guidance standards are focused on the sum of 11 specified PFAS compounds to less than 90. That includes PFBA, which is fairly notorious for being the most difficult compound to remove with existing technologies. Denmark is similar with 12, sum of 12 PFAS compounds, less than 100 parts per trillion. And if you look at Australia, they're all the way down to 0.00023 ppb, or less than a part per trillion for PFAS. And that's not for human health. That's for actually the ecosystem, protecting freshwater environments and the organisms therein. Other things going on in Europe, the European Food Safety Authority and Drinking Water Directive. This is really interesting, proposing strict limits for 20 compounds, and in a year and a half, January 2023, all EU countries must turn these into rules that are enforced. Individual countries can have even more stringent limits. And for now, drinking water is the primary target, but that's changing. We're seeing wastewater also being added around the globe, stormwater, soil, and groundwater. So lots happening. The next slide, we're going to turn the discussion to proven technologies for water treatment. And it's a bit of a review from previous presentations I've given, but I think it's important when you talk about piloting to understand what the options are to pilot for technologies. The incumbent technology, carbon, has been around for decades, and people are comfortable with it. I have a column in my basement for radon and manganese removal, and you know it has its place, and we're going to talk about what that place is a little bit here in the coming slides. RO and nano are use pressure to force water through a semi-permeable membrane. Those technologies are gaining traction. We'll talk about that a little bit more. Foam fractionation, relatively new, but it has its place. These compounds want to foam. They are surfactants, so in a lot of cases it makes sense to do this in a very cost-effective way to remove the compounds from the water matrix. And last but not least, ion exchange resin is really gaining in popularity, both single-use and regenerable, and that's largely due to the very high capacity because of the dual mechanism of adsorption and ion exchange that's at play. So granular activated carbon, being the incumbent technology, focuses on adsorption. That is the mechanism at play. Van der Waals forces are the attractive mechanism. The hydrophobic tails on these PFAS compounds don't fit well in the polar water matrix, so they tend to instead achieve a lower state of free energy by adsorbing onto the carbon. This works well for the long chains. Over on the right, you see the typical lead lag vessel configuration. Short chains, not so much. So, you know, we look at other technologies like reverse osmosis, which tends to do a nice job on the short chains. Sometimes nanofiltration can even be used. The challenge with these membrane technologies is they always generate a reject stream, which is typically going to be somewhere between 10 to 25% of the forward flow. So it's sizable, and so it can be as low as 5% depending on how many stages there are. But the bottom line is we need to deal with the reject, so it can often be used as a step prior to GAC and or ion exchange resin. So again, you know, we're forcing water through that semi-permeable membrane, and I think we're going to see more applications of reverse osmosis in the future. Foam fractionation, probably most of you haven't seen a foam fractionation system at work. Our company is starting to use it more frequently, typically as a pre-treatment step. Basically, the way that it works is you have the dirty water coming in the top of the vessel, flowing down through the vessel, and clean water coming out the bottom. This flows counter current to the air bubbles, which are introduced at the bottom of the vessel with a lot of turbulence. That whips the PFAS compounds into a froth. They form foam, which of course floats to the top and then can be skimmed off. So we're talking about largely plastic components, inexpensive, simple technology, very little, if anything, to clog, just that counter current flow of water and air. It's a good pre-treatment step. It's good on nasty waters like landfill leachate, where you plug up just about anything else, whether it be a membrane or carbon or resin. Works well when you have high TDS, like high chloride concentrations, for instance, because it tends to be agnostic to chlorides. So definitely a place for foam fractionation now and in the future. Ion exchange resin really gaining in popularity because of that dual mechanism. The adsorption and ion exchange, we get rapid kinetics and higher capacity. We have a mock-up of a resin bead over on the right. I mean, those blue dots are positively charged sites, which is where the exchange takes place. There are literally many, many, many thousands of those sites on a 1 millimeter bead. So this is obviously a simplified version. But if we look at the next slide, this gives you an idea of how that typical PFAS compound is separated into its hydrophobic fluorinated carbon tail, and then it's typically a sulfonate or a carboxylate, which has a negative charge and is hydrophilic or water-loving, At the other end, that's where the exchange takes place. The adsorption is on the hydrophobic tail. Opposites attract, so that's why that negative head is attracted to the positive charge on the resin site. What does that mean? Well, it means if exchange happens a lot faster than adsorption, then resin systems are going to be smaller than carbon systems, and they are. And this gives you an idea of that stark difference in footprint. And even with this difference in size, the resin vessels tend to be changed out or regenerated less frequently than the carbon in those carbon vessels. So some real distinct advantages over carbon. And how do we make it more sustainable? Well, our company has developed and patented a process to regenerate ion exchange resin on site in vessel. And all these technologies typically have some level of pretreatment before them other than possibly foam fractionation. After that, just like carbon or single-use resin, we have lead lag ion exchange resin. Maybe there's going to be a polished resin depending on if we're going after short chains too. But what we've developed is a solution, is a blend of alcohol and brine or salt that once that lead vessel is saturated with PFAS, it can be taken offline and regenerated with this regenerate solution, which washes, if you will, the PFAS compounds back off. They're collected, sent through a distillation process, which recovers the regenerate solution for reuse. And what we're left with is a very small quantity of liquid brine with very high PFAS concentrations. We also have developed and patented a process called superloading that then further reduces that volume by about another order of magnitude into a solid waste. So just round numbers, these series of concentration steps take, just by way of example, this would be typical, 1 million gallons of fairly heavily contaminated, PFAS-contaminated water. And after the removal and the regeneration and the recovery, the distillation, the superloading, that 1 million gallons boils down, if you will, to less than a gallon of solid waste. And so very sustainable, less trucking, less burning, less burying, and less liability associated with this process. Especially, it's really applicable to those sites where the incoming concentrations are relatively high in the ppb levels as opposed to the ppt levels. Because if you have a drinking water that you're treating ppt levels, you may only have to change the resin once every year or two, and in that case, it's hard to pay off a regen system. But there are a lot of industrial wastewaters and leachates and contaminated groundwaters that have much higher concentrations than that. Okay, going to shift gears again here and get into the pilot studies and the importance. What are the elements of a good pilot study? And it has to be designed well, right? And that starts with an accurate design basis. What does that mean? Well, we need to understand the properties of the water to be treated. It's not as simple as just knowing PFAS compounds and their concentration. What else is in the water? How much iron? How much natural organic matter, as measured typically by TOC? What about TDS, chlorides? All kinds of, it could be colloidal compounds, silica. We need to understand and have a good grasp on the water that we're going to be treating, and that helps drive understanding pretreatment. We also need to understand what's the desired outcome. Are we just trying to remove PFAS, PFOA to less than 70 ppb? Or do we need to remove some of the short chains too? And if so, which ones? What about precursors? Pretreatment, we're going to get into some examples of that, but extremely important to pay attention to that. What are the site requirements? Where are we getting the water from? Is that source representative? Is it coming from a pressure main or a deep well or a break tank? Or do we need a break tank? Where's the treated water going to be discharged to? And here's one. The hydrogeo considerations are often not given enough attention or all out neglected. And what happens is there'll be some low flow sampling that takes place from these monitoring wells that have been sitting around for years. Maybe there's been some sort of pump test, but oftentimes not enough. And what we get, turning to the next slide, is on the left is an example of a design basis that was given to us, and the concentrations are shown. I know you can't read them, but up in that upper right, there were concentrations in the source areas up over a milligram per liter, or ppm, and in some cases higher than 10 ppm. And so that was the design basis that we were given. We weren't given a chance to run a pilot. The timeline of the project was accelerated. There was no pump down test. And so over on the right, what you see is a pilot scale foam fractionator. We were running this test and had started this earlier at another site to understand if this might be a good pretreatment step for sites that have really high, ppm type levels of PFAS. And so bottom line, based on the design basis, a foam fractionation system was installed for pretreatment, but was never used, because once the extraction wells started up and moving water through the source zone and through the aquifer, the levels dropped by 2 orders of magnitude, dropped 100-fold. And so, and that was supposed to be the foam fractionation system's job, was to drop the levels by about 100-fold. It basically 90%, and or even more than that, and just it didn't come to fruition. It wasn't needed because that pump down test was not performed up front. So some good lessons learned for us there to demand that the proper pump down test be done to give you an accurate view of what the concentrations are going to be when your full-scale system starts up and starts moving water through the aquifer and the source zone. Don't forget pretreatment. I mean, the photo on the right shows a typical setup for us, where we have a sand filter, we have pretreatment carbon, pretreatment resin, and you can see that doesn't look like sand because it's removing a bunch of junk from whatever water source this is, as is the carbon, as is the pretreatment resin. But you see those two resins on the right, they're doing the heavy lifting on PFAS removal, and they're staying clean. So I think it's a good visual example of what pretreatment can do for you if done right. And so we need to understand, like, what are we looking to pretreat? Is it suspended solids in iron? What state is the iron in? Will sand filters or bag cartridge filters work? Will they be representative? Is there colloidal fouling matter that is going to need to be coagulated and flocculated and settled out? Are there metals that are going to need to be precipitated? What about any biofouling? Carbon, backwashable carbon, is good for removing solids and TOC. There are lots of specialized resins. We've used them to remove iron and natural organic matter. Some metals can be effectively sequestered or handled with pH adjustment. Biocides or organic dispersants can be used to control and prevent biofouling. Oxidation, advanced oxidation, can be used to convert precursors ahead of time, so these compounds can be removed by your system. The next slide shows just some examples. The top picture is a pilot nanofiltration system that we used to remove. Actually, what we're doing here was we were removing PFAS, but letting the chloride flow through, so that when we got to the next step, which was anion exchange resin, the capacity of the resin was improved significantly, because the chloride was no longer with the PFAS. Right, the concentrations of chloride were greatly reduced in relation to the PFAS, and the volume was less, so we could use smaller vessels. The bottom group of photos shows an industrial wastewater that had a colloidal suspension that was certainly going to foul our downstream reverse osmosis and anion exchange resin system. If you look at that photo on the right, the beaker on the left, we started in the lab with bench testing, took those learnings from the lab because we found the winning combination of ferric chloride worked and ferric sulfate worked. Both needed some pH control to achieve the optimum pH. The next slide shows what that looks like. The pilot system is currently being deployed. It hasn't started up yet, but that pilot system was designed and constructed based on the results of that basically quick and dirty bench testing that took place to find the right coagulant, the right mixing conditions for the rapid mix and the flock tanks. And you can see here we're using a lamella clarifier. So we're adding ferric, we're adding caustic, we're mixing, we're flocking, we're settling, we have sludge storage and thickening tank. And that's all based on some good work done by our R&D folks in the lab to nail down how are we going to remove these solids that we're going to foul the downstream UFRO and resin systems. It's all about the data, and the importance of data collection is often underestimated. You can't rely on the client, even if they say they're going to do it, to run your pilot. Very selective cases can that work out. These pilots are substantial investments, and we need to give them the care and attention they deserve. And my mantra, bad data, worse than no data. And if you go to conferences, there are a lot of bad pilot studies out there that people present on. So you need to plan the test, run the test, and do it right. When we run a pilot, our operators put out daily summary emails that the grader team reads. We have daily support calls to talk to the operators and make sure they're comfortable with the way things are operating and the data that's being collected. And another thing, you can't be afraid to hold samples. Don't feel like you have to send every sample in that you collect. It's a lot more cost effective to stash some in the fridge or the freezer. There's no scientific basis for a 14-day hold time for these forever chemicals. If there was, we'd put them in the fridge for 14 days and we'd have our problem solved, right? So to hold those samples and save a bunch of money. In some cases, the regulators involved, and it gets a little tricky, and you need to adhere to the 14-day hold time because these are deemed for compliance, but a lot of times pilot tests are for information and not for compliance. So we need to take advantage of that and common sense. And once on paper, the data set takes on a life of its own, so we need to collect good data. The next slide shows an example sampling and analysis plan. The yellow are PFAS and wet chemistry. The blue are hold samples. So in other words, when the yellow samples are collected from various points in the pilot, they are sent into the lab immediately. The blue samples are stashed in the fridge and only submitted if necessary based on the upstream results. Moving on to scale, very important to pay attention to scale. And that all depends on what process or processes you're going to be piloting. Resin and GAC columns tend to scale up well and don't typically need to be bigger than 2-inch diameter columns. Some of the solids removal, the coagulation, flocculation processes need to be scaled up for the pilot test. Same with some of the chemical processes. And we just need to understand what we're doing in the way of treatment and how well those processes scale. Do you need a sand filter? Can you use a bag or a cartridge filter to simulate? Well, it depends. If you're using a regenerable resin process, are you going to have enough spent regenerant solution to do the follow-on testing like superloading or on-site destruction? So often the question is, how big is big enough for that pilot test? And a lot of that depends on how big does it need to be to get the client and the regulators on board. because you hate to spend a bunch of time and money running a pilot test only to have it be rebuffed by the regulators, for instance, because they don't think it was a large enough scale to be representative. Next slide shows an example, a couple of examples actually. On the left, this was a single-use resin pilot. You can see these columns are not very big. That scaled very well and was just fine for the purposes of understanding what empty bed contact time should be used and how long we would expect to run between resin changeouts. Over on the right, you see that the vessels are a lot bigger. You know, these are something like 12-gallon vessels, and I think they're like 9 inches in 9 or 10 inches in diameter, typical of what you have in your basement if you had a water softener. So much bigger, treating a lot more water. In this case, it's a regenerable resin system, and we needed to be able to generate enough still bottoms from the regen and recovery processes to be able to test the superloading process as well as on-site destruction. Clarkson's DMAX plasma destruction system was the one that was tested in this case. Okay, on to the case studies. First one I'm going to talk about took place in Australia. This was a few years ago, 2017-18 timeframe. We started up a system that was designed to remove PFAS from stormwater that was flowing off-site from a defense Air Force base. And groundwater was actually exfiltrating into these stormwater swales, so the iron was reduced, and then it would become surface water in the stormwater swale, get oxidized, and then start precipitating. It was also complexing with natural organic matter in the water. So we'd get a big rainstorm, and all of a sudden we'd have all this nasty, you see on the right, that photo on the right, it was really fouling up the system when we first started up our full-scale system. So we quickly enacted, set into motion a side-by-side pilot scale system in the very shipping container that was holding a lot of our full-scale equipment, and tested 18 different media to see if we could find one that would be effective at removing the majority of that iron organic matter complex. And the clear winner was identified and successfully implemented at full scale. And on the next slide, it shows that column on the right. That resin is very white when it's new. This is downflow pilot test. You can see that there's that dark color, real dark brown is natural organic matter. The more orangey color is iron. And if you look at the plot over on the left, that black data line represents the influent to this, or to the 18 different media that were tested. And that was taken from the sand filter effluent of the full-scale system. The orange data represent a pretreatment media that we call PT1A. The gray over on the left represents a different, in this case it was a Pure Light product, and the Pure Light makes a lot of good products. In this case, it didn't perform as well as the one in orange, and you can see that backwashing helped there in the middle, and over on the right, giving it a brine regen restored almost all of its capacity. So that resin has been working in place with periodic cleanings for years now, protecting the downstream PFAS removal system, which is a different type of resin. And periodically restoring capacity with the brine regen, and sometimes a little more aggressive regen on not very frequent, on infrequent basis. But the cool thing is, we started up a system. We had a problem that we didn't see coming. We immediately went to a side-by-side pilot test, solved the problem very quickly, and implemented it very quickly and problem problem solved. Over on the next slide, this was a side-by-side test. Excuse me, grab a drink of water here. Of a single-use ion exchange resin versus a carbon, and it's a New England public drinking water supply. The inlet PFAS in this case was greater than 2,500 ppt, or 2.5 micrograms per liter, not your typical drinking water well. And what was done here, a lot of things were done right. The data were collected successfully. Attention was closely paid to the pilot. Daily discussions, daily report outs. An independent consultant was hired to help design the test and interpret the data to develop a life cycle cost analysis, which is the only way to really get to the bottom of what is the right technology, right? If you need to run a pilot test, which we often do because this is relatively new contaminant, new technology, there's nothing like doing it side by side, collecting the right data to run that comparative life cycle cost analysis. These are some of the breakthrough curves for that test. We see carbon on the left, resin on the right. The black dotted line is the influent PFAS plus PFOA, and you can see that over on the left, an empty bed contact time of 10 minutes is needed to get any reasonable capacity out of the resin at this high, 2,500 part per trillion influent level. But you notice the resin on the right went for over a year, and the smallest vessel, 1/4 the size of the one that worked okay in carbon, 2.5 minute vessel lasted for over a year. So definite advantage carbon there. On the next slide, we also looked at the short chains. Interesting that PFBA broke through carbon at first higher than the influent. That's called chromatographic displacement. And then over on the right, more classic breakthrough curves of the 2.5 to 5, 7.5, and 10-minute columns. Next case study. This was an interesting one. Really interesting because it wasn't just PFAS. It was a lagoon that was collecting de-icing fluid, propylene glycol. It was filled with iron bacteria and hundreds of ppm, sometimes I think even more, of propylene glycol, which is extremely biodegradable. And we knew starting out this one, we were going to run a side-by-side pilot to stay ahead of things. We started it ahead of time, and we used it as our canary in the coal mine to figure out if we were going to have to do anything, especially to address the biofouling concerns that we knew were going to come with the active biogrowth that was going to happen on our media due to that highly biodegradable glycol solution. So we also coordinated with our R&D lab to find the solution to the biofouling and get ahead of it. And it turns out that if you look over on the right, that the black growth that you see in that column, that resin column, is the beginning of a very substantial colony of bacteria that are chowing on the propylene glycol coming from the lagoon. Because basically they're collecting de-icing fluid off the runway, but they also do fire training, so there's a bunch of PFAS in there too. So our R&D folks in the lab they looked at a bunch of different biodispersants and found one that not only works very well in preventing the bacterial growth, but also could be, the destruction could be catalyzed simply by having a small vessel of carbon at the tail end of treatment. And yeah, it also, this particular one didn't impact the resin's capacity. Some of them did actually used up sites on the resin. So it was a real victory in the R&D lab that was translated to the pilot scale to prove out on site, which was running ahead chronologically of the full-scale system that you also see down in the bottom middle. And so a really cool project, very successful. And 2 other entities had been in there before us and failed. And, you know, our approach to piloting won the day there. And we're still on site, you know, a year and a half later. And this plot just shows the outstanding results. The influent and the effluent been consistently non-detect for a year and a half, below 1 part per trillion for PFAS and PFOA, this is treated waters being discharged to the municipal sewer and flowing to the POTW. The next slide shows pilot test at an Air Force base in the Midwestern United States. The idea here was to not only look at regenerable resin, but also to look at destruction of the still bottoms from the regeneration process. Excuse me. So we teamed up with AECOM on this one and their defluoroelectrochemical oxidation process. And so over on the left, you see inside a shipping container a resin system with a sand filter pretreatment system with a tank to hold and thicken the solids from backwashing the sand filter. Over on the right, you see that resin regeneration system and regenerate solution recovery system, complete with distillation to take the high volume, low concentration PFAS in the groundwater and turn it into very low volume, very high concentration solution that could then be destroyed using, here on the next slide, the defluoro process developed at the University of Georgia and in conjunction with AECOM. And yeah, so that defluoro process couples very well with ECT2's regenerable resin process. And so this was a really, really cool pilot study. This next slide just shows a system. We're starting to do a bunch of testing in Sweden. Sweden's really picking up steam on the PFAS front, not just drinking water, but also groundwater remediation, landfill leachate, wastewater. And so again, you know, just some typical data you see over on the right, where the resin is lasting a lot longer than the carbon. The next slide just shows a couple other pilots being performed by our team in Sweden. The next slide shows another public drinking water supply pilot. The interesting thing here is that we really need to speed up the test. I mean, one of the biggest challenges with pilot testing, even column testing in the lab, resin for PFAS removal, it can take longer than a year to collect enough data to show breakthrough, and that's untenable. So what we did here was typically you want 30 to 36 inches of total bed depth, but we did 4 columns in series. For the resin, they were each 9-inch resin bed depth, so 9 times 4, 36-inch total bed depth. And, you know, we kind of took a flyer on this one, but it worked really well. And so you see the 3 different sets of columns, 2 different resins and 1 carbon. And then here are the data on the top. Red, that's the influent. The orange line in the middle is the GAC lead vessel breaking through. The next column to break through in the dark blue was the GAC lag. And then the resin is just chugging away, you know, after almost a year's worth of data collection and a lot of gallons and bed volumes being processed. So this is my last slide. I wanted to finish at 2:45, so I think we're pretty close. I want to save plenty of time for questions, but I do want to take a little bit of time on this slide because the take-home messages are interesting, compelling, and important. And we have to keep an eye on the regulations, right? You need to know what's going on in the state that you're working in or the country that you're working in. They're evolving. They're only going in one direction, and that's down. And laws are actually being enacted and enforced. We're having 20 compounds now instead of 2. It's really an evolving landscape. yeah, more compounds being added. The concentrations, the limits starting to dip down, drive down into the single-digit nanogram per liter or PPT level. You know, when you think about what technologies to pilot, there are really 5 proven ones out there, and you're each going to get a copy of this, PDF of this, as a follow-up. And so a lot of good info in here, and if you need more, feel free to reach out to us, and we're happy to provide more or answer more questions, especially if you have a specific application. Don't cheap out on the pilot study design, especially hydrogeo. Get out there, run that pump test, get water flowing through the aquifer, through that source zone, so the samples that you're collecting are going to be representative of what you're going to be running in the pilot, and the pilot that you're running is going to be collecting water that's going to be representative of full scale. Because I say it over and over again, bad data is worse than no data, and we really need to collect, execute, follow through, run the test yourself. Your company should run it. You should have somebody reporting out every day. You should have people on the phone supporting, you know, routine check-in calls, meetings, support on site, and the pretreatment. I just cannot emphasize enough how important it is to get the right pretreatment or pretreatment steps, combinations of pretreatment, because these resins, these membranes, they clog very easily, and we need to get the foulants out so they can do their job and remove PFAS. And please understand that it's okay to collect more samples than you think you're going to need and hold them, because holding samples has saved us many times. Also sending dupes in to different labs sometimes. These their LC mass specs, they get fouled up, and you know, it can take a while for these labs to start collecting good data again. It's good to have a backup lab, at least one backup lab, to be double checking and sending some blind dupes. And then, you know, make sure you run that pilot at a scale that is going to gain buy-in from your client and from the regulators, from the consultant. Really important. So here's my contact information. Here's one of our employees setting up a pilot test, getting ready to start it up in a couple of days. And we run a lot of pilots. I think we figured out how to do it. Learned a lot of lessons the right way, a lot the hard way, and really important to do it right. So hopefully I've imparted some valuable knowledge here, and I'm going to be quiet now and hand it back to Jessica, and we'll get some questions flowing.
Sounds good. Thank you very much, Stephen, and I appreciate the fact that you ended the presentation with some best practices as well as advice for everyone that is on today. I think that's a great way to end the event, at least you're part of the event, before we jump into the Q&A. So we have about 10 minutes or so for Q&A. Again, keep submitting questions. We might not get to your question over the next 10 minutes or so, but I know Steve or a member of his team will get back to you as soon as possible, especially those who have been asking very, very specific questions. All right, so let's get started. Steve, are there any biological processes that prove to work for PFAS or PFAS treatment?
There's a lot of work being done to find a biological solution, and some of the precursors will readily biodegrade to PFAS compounds or other PFAS compounds. But in terms of breaking down compounds biologically, PFAS and PFOA, there has been some progress, but the carbon-fluorine bond is the strongest covalent bond known in nature, and it is extremely difficult to degrade, even with under extreme conditions of heat and strong oxidizing chemicals, advanced oxidation. So I think we're a ways off from having any real biological solution that is going to find any sort of widespread application.
Thank you, and I'm sure that was a question that others had as well. This next question, I'm going to have you go back to something that you talked about earlier. Pilot duration is a significant limitation. So can you kind of repeat your comments on how to overcome this?
Yeah, it's a tricky one. I mean, what I didn't mention is there's a lot of work going on to try to find an ACT, an accelerated column test, or an RSSCT, rapid small-scale column test. And what those are much smaller columns with ground-up media, and it just they have some useful qualities. They have their place, but we really need to caveat the results, because even the large carbon companies will tell you. I sat through a presentation by one of them recently that you have to use great care in interpreting the results. So what else can you do? Well, like I showed, you can use smaller columns and shorter bed depths and chain them together in series. And so that test that we did on that one drinking water supply in the East Coast, we had PPT levels like 150, 200 parts per trillion in the effluent. So we knew it was going to take a while to achieve breakthrough, especially through the resin, which is why we used such shallow, 9-inch bed depth. Seems ridiculous, but you piece 4 of them together in series, and essentially what you end up with is a 36-inch bed depth, and it allows you to look and see if you're getting some reasonable data at those intermediate points. So there are a couple of different options for trying to speed things up. You can stick with the 2-inch columns and just go with shorter ones in series, or you can attempt to run an accelerated column test or an RSSCT. But I advise great caution in interpreting the results. They can be used to sometimes compare like different resins or resin to carbon and those sorts of things, but they often greatly overpredict how long the resin or the carbon is going to last between changeouts. And I think a lot of that has to do with the grinding of the material and eliminating that or taking significant strides, measures at reducing the impact of diffusion to get into the interior of the bead or of the granule.
And our next question, and thank you for going over that answer again, and our next question actually kind of piggybacks onto what you just talked about with the RSSCT, and you did answer a little bit of it in the question, but what is your experience with using the rapid small-scale column test to shorten the time required to perform a lab or pilot evaluation?
Yeah, we actually have a fair amount of experience, and we've worked with some of the universities on the development of that testing and shared data and shared methods and whatnot. It's still a test that's under development, and we have used it. We've shared the data. We have cautioned users of the data to use it with care, and in some cases we think it provides a good or a reasonably good comparison of which product is going to work better for this water under the conditions that they expect to see at full scale, whether it be a resin or a carbon or some other type of absorptive media. But in terms of trying to extrapolate that data to estimate how long a full-scale system, resin bed or carbon bed, is going to last, that's, we're talking, these RSSCT columns are like the diameter of a pen or a pencil. So that's where scale becomes a real issue, and I think there's still a fair amount of work. I know there's still a fair amount of work to be done to glean a ton of real useful information. They do have their place. We just need to be careful.
Which definitely makes sense, and I do appreciate the level of detail that you're going into with your answers today. I know our audience appreciates it too. The next question, and we have time for maybe a couple more after this. This is about ion exchange regeneration. It's kind of a two-part question. So is the ion exchange regeneration primarily an aggressive backwash, and how complete is the regeneration process? So like a typical percentage.
Yeah, it's a really good question. It's not a backwash. It's a fixed bed, so the bed does not expand. It's a chemical process whereby the alcohol in the regenerant solution dislodges the hydrophobic tails of the compounds, and then elevated chloride concentrations in the brine solution dislodge those charged anionic heads. And so, we'll lose a little bit of capacity after the first regen, maybe a percentage or two, but the data to date have shown that we can do, in Australia, for instance, we've done dozens of regens without losing noticeable capacity after that first one, where you'll lose maybe 2-3 percentage points. So it does a great job. It's easier to knock the short chains off. You get the longer chains off, like PFAS and PFOA, takes more bed volumes, but still it's very efficient and effective process to chemically remove those compounds.
Great, thank you. This question has to do specifically with the company. Is EC2 also involved in remediation of PFAS contaminated soil and or sediment? We do have some partners that we're working with on soil and sediment removal. It's not really, I wouldn't say it's our bread and butter, but one of the things that we're doing is helping our partners figure out how to clean the soil, right? And since we figured out how to get PFAS off of resin, which is about the toughest thing to get PFAS off of because of that dual mechanism of attraction, we have had success in helping to use a similar regeneration process to clean soil and sediment. So it's not our strongest suit, but like I said, we are involved and we are helping some of our partners in achieving some good results. I'm going to jump in real quick and ask you a question. What has been the biggest change you think at ECT2 since you have joined the company, or how have you seen this company evolve as regulations change and as the systems around you have changed?
Yeah, I think what I've seen change is number one, there's a lot more focus on the short chains, and at first it seemed to be like all PFAS and PFOA in the States and PFAS and PFHXS in Australia, and branching out into some of the shorter chains in Sweden. But as time has rolled on, a lot of the challenges presented to us are more along the lines of getting the short chains off, doing so in conditions of high TDS. One of the things we've done is we've moved, in addition to all the groundwater remediation and drinking water treatment, we're doing a lot of industrial wastewater treatment, and that opens up a whole new can of worms with a bunch of different compounds, a lot of them short chains. I guess another thing that has changed is we're operating a lot of the sites. There's a lot of our clients like the work that we're doing, and that they want their investment to be maintained and operated and managed appropriately, so they have good results, and we have a flawless track record there. I mean, our systems have zero exceedances around the globe. So we've become kind of a soup to nuts technology company that we develop new technologies, we patent them, we commercialize them, we deploy them, We oversee the construction. Sometimes we're the lead on the design build. We're involved in startup, and now we're also operating well more than half of the systems that we're installing. And the growth has been pretty phenomenal too. Hiring two people a week on average these days just to keep up with demand.
Wow, that's impressive and good for the company and different clients that you work with. We are pretty much out of time, but I think it's a good idea to end with this last question, and then we'll wrap it up. Can you speak a little bit more to the water quality conditions that trigger foam fractionation as a pretreatment step? We had a bunch of questions come in about pretreatment.
Definitely. I see foam fractionation as being effective when you have, as a pretreatment step, when you have super high, like 10 to 100 or more milligrams per liter, ppm levels of PFAS. It's a great way to knock that, 98, 99% of the PFAS off. So now you've got a reasonable amount going to your downstream resin or carbon process. Another case where I think it has great value is when we've got some short chains, so carbon doesn't work well, but the resin doesn't work well either because we have high TDS, especially high chloride concentrations. We've used foam fractionation very effectively in those situations. And I think the third I would highlight is landfill leachate. It's just nasty, and it will plug membranes, it'll plug carbon, it'll plug resin, but foam fractionation, leachates love to foam, and so whenever you're looking at treating a landfill leachate, foam fractionation should be included as one of the technologies to pilot.
Great. Well, thank you for wrapping it up with that question, and thank you, attendees, for staying on a little bit past the hour. Thank you, Steve, and for the entire team at ECT2 and Montrose Environmental Group for all the work you put into the presentation today. Again, attendees joining live or on demand, I have now made the slide deck accessible, so it's just going to take about a half an hour or so, and then you'll be able to log back into this link and access the slide deck. At this point, we are going to end today's webinar, and I hope everyone has a wonderful rest of the day. Thank you again, Steve.
Thank you, Jessica. Appreciate it. And thanks to the audience, too.